So then I also want to acknowledge the organizing committee for the opportunity to talk, finally to get here and do this. Okay. So let me see, let me move this so I can see. All right. So I have ventured into the field of reproductive biology. So this talk is going to be on male factor infertility. I'm new to this field. I was surprised, I guess, to see that male factor infertility is a large factor. So there's a lot of space for improvement and further understanding of this condition. Because most of the causes are due to unknown spermatogenic failure, so developing sperm cells. And then there's this whole class of just totally unknown reasons for infertility. But the male is a factor in a large proportion of couples that are having a problem having a child. Initially, so I got into the center of this talk is this Mier3 gene. It's not a microRNA. It's a family of 3 genes. And it originally got into the Mier3 business through, it's a GWAS, human GWAS and rat QTL nominated candidate gene for breast cancer susceptibility. And relevant on this slide for this talk is that expression of this gene is positively correlated with rat mammary carcinoma susceptibility. So we developed resistant and susceptible congenic strains, pinpointed Mier3 through this expression. So colleagues at the University of Wisconsin, namely Kathy Krentz and Dustin Rubinson, went in and made targeted mutations. And we did see that targeted mutation of the gene decreased mammary cancer susceptibility as we hypothesized it would. All right, so here's, for those that are interested, what I think this is one of the first times that Kathy used electroporation of Sprague-Dawley embryos. And she put 94 embryos into 2 recips, and she ended up with 13 live pups with edits, right, out of 15. So low starting points, but good yield. And then there were 4 rats that had premature stop codons, and we focused on those. I keep going to the screen to move. Here's a look at the sequence relative to the PAM site of the 4 mutations we selected to advance as founders. And you can see the deletion and insertions here. And then the predicted effect of each of these in comparison to the reference on the terminal amino acid sequence. So the red amino acids are introduced by the indels or the deletions up here, and then the relevant stop codon here at the end. So you end up with what we predict to be truncated proteins. Okay, we first noticed a fertility problem in males when I tried to fix the strains. And you can see here that I set up 11 homozygous targeted mutation males, and only one of these gave me a litter. And it was a litter of 3 pups, and I set that male up again, and it didn't produce a litter. In contrast, the hets seem to have normal fertility. So at least we can keep the line going with hets. And the females, contrary to the males, the homozygous females appear to be no apparent fertility problems. They give live births, they lactate all the way to when the pups are weaned. Okay. Mier3 is expressed in testis tissue. It's most highly expressed in the seminiferous tubules, so that's the place where spermatogenesis occurs. Ductal structures, you'll see those at the end of this talk. And then it's also highly expressed in Leydig cells, which are the cells responsible for producing testosterone in the testes. Okay, so some data here. We compared testis mass and area. And doing this in 2 different age groups, we see that the homozygous targeted mutation animals have lower testis weight compared to both the reference, so the homozygous for the reference allele or the Sprague-Dawley Mier3, and then also to the het. and then area. So smaller gonadal size in the targeted mutation homozygous males. Epididymis lengths. So then the next thing we wanted to do is see if there was a difference in sperm concentration. So we took epididymal sperm out and measured the length of the epididymis in these animals, and indeed these were shorter, and sperm concentration was affected as well. We see lower sperm concentration compared to the heterozygous animals. There's quite a bit of variation, it appears, in the hets, but it was lower concentration. Well, one of the three animals over here actually had zero sperm cells in their epididymis. Here's histology, comparing the homozygous reference allele animals. I pulled some of these out. These are at 40x compared to the recessive targeted mutation. You can see, let's walk you through here. This will be a test for me as well. as a reproductive biologist, I'm not. So here you have the ductal structure. I'm familiar with that. You have what's going on is you have the germ cells, spermatogonium starting here as diploid, and then as they progress through divisions, they become haploid, of course, and then eventually into spermatozoa. You can see the spermatozoa in the lumen of this duct here. Here, outside of the ductal structures are where the Leydig cells are. They are the cells that produce testosterone. And then within the ducts, you have cells called Sertoli cells. And my understanding of these is they're not completely well understood, but the working hypothesis and that is that they're there to support the progressing and dividing germ cells as they enter into the lumen. Now, I haven't quantified or anything this, but visually we can see clearly that we know that the targeted mutation homozygous animals make spermatozoa. So you can see some ductals in here that are progressing through this, but we see a lot more less structured morphology. And then we see a lot of these ductals that are filled with maybe something that looks like fibrotic tissue, and then the lumens contain no spermatozoa. So we get more variation over here, and we don't quite know the end result of all this. But you can see this ductal and this targeted mutation animal that looks like it's functional, making spermatozoa. You can see that the Sertoli cells are here and look intact. But then if you look in some of these other, it's difficult to see any Sertoli cells. We haven't done any staining for that cell type, but you can clearly see visually that they seem to be missing. So that could be one direction that we go in with this. And then the overall structure is different. And then hopefully your eyesight, your hematoxylin vision is better than mine to tell the difference between the pink and the blue. Right? So. Okay, so wrap this up. Here's a summary. So we saw lower fecundity in the males, but no effect, apparent effect on females. It appears to be something going on in the seminiferous ductals of the testis, where there might be a hypothesized problem with spermatogenesis. And we can see these visual differences in the tubules and effects. We can clearly see, I didn't show you any movies. I don't have those data properly analyzed yet, but you can clearly see if you look at the movies, morphology, abnormalities, and then also motility issues with the sperm here. And then I'll just leave you. I understand we're going for coffee, so maybe something to talk about. Speculation is if this is evidence for an intralocus sexual conflict. Because in the females, we see less, the hypothesis, the working model is less expression of this Mier3 gene is protective towards developing breast cancer or mammary cancer in the rat. And then possibly the flip side of that is less expression of this could induce fertility problems for males. So, all right. Thank you.